Thank you. I want to thank the organizers for inviting me to share my work with you. My name is Rulla Tamimi. I'm a cancer epidemiologist. I'm a professor of population and health sciences at Weill Cornell Medicine. And I'll be talking today about the integration of molecular and radiologic features as a way to really understand breast cancer risk. And we'll discuss some of the implications for risk assessment and prevention. I have no financial disclosures. So in terms of learning objectives, I have 3 learning objectives for today. The first is to understand the importance of thinking about the life course and considering timing of exposures. I think this is important for all cancers, but especially for breast cancer. I'm going to talk a little bit about mammographic density, and our goal is to really understand how mammographic density is a risk factor for breast cancer. And then finally, really to understand how integrative epidemiology can help us to understand how risk factors influence disease, and specifically today, cancer. So the overview of my talk, my entire talk is about breast cancer. So I'm going to start with some descriptive epidemiology. I'm going to also give a little bit of background on the nurses' health studies. These are the epidemiologic study that'll be the foundation really for 2 of the recent works that I'll highlight. And then I'll launch into 3 areas of work. The first, looking at early life body size as a risk factor for breast cancer. The second, looking at mammographic density as a risk factor for breast cancer. And then in the third one, this will be in a different population-based study, but looking at the impact of vitamin D supplementation on breast tissue. And then I'll close with some final conclusions and future directions. So in terms of descriptive epidemiology, as many of you know, breast cancer is the #1 cause of cancer in women in the United States and globally. And there are over 280,000 new cases of invasive breast cancer diagnosed in the US each year. It's also the 2nd most common cause of cancer death in the United States. This is second only to lung cancer, with over 43,000 breast cancer deaths each year in the United States. I like to show this graph. This is a graph that shows incidence rates and mortality rates in different countries, those of high incidence and those of low incidence. And if we look at the brighter pink bar, I think what you can see is that there's about a 5-fold variation in terms of incidence rates between high incidence countries and low incidence countries. And some of this may be due to detection, screening that's able to detect more cancers. There's also really the belief that there are different risk factors and the uptake of different risk factors across these different populations that influence incidence rates. And so this gives us some idea that there may be modifiable risk factors or things that we can learn to really think about how can we change some of our behaviors or exposures in order to reduce the risk of breast cancer. In this graph, we are looking at the incidence rates of breast cancer in the United States over time. And we'll just focus on this bright pink bar, or the bright pink, the top line there. And I think it's important to look at the trends over time to really see what is the burden of disease in our population. And you can see that in the 1980s, there was a wide increase, or there's a large increase in the incidence rates of breast cancer. And this is really due to the uptake of mammographic screening. So with increased screening, we detected more cancers. We also see a continuing trend of increase in breast cancer, even between 1980s and 2000. And these are really thought to be attributable to risk factors that have been changing in the population. So in general, women have been delaying childbirth. Women have been having fewer children. There have been more hormonal exposures, both with oral contraceptives and postmenopausal hormone use. All of these are risk factors and are really acting in a way to increase the risk of breast cancer in our population. In addition to that, we've experienced the obesity epidemic, and we know that postmenopausal obesity is a risk factor for breast cancer. There was a decline around 2001, and this is really attributable to, there was a publication that was published in 2001 from a randomized control trial showing that women who were on hormone therapy and estrogen plus progesterone hormone therapy were at an increased risk of breast cancer. And a number of women had been taking these drugs, really a lot of them for the thought that it could reduce cardiovascular disease risk. And a number of women discontinued use, and you can see that turned into a demonstrable decline in incidence rates. But really, since the 2000s or early 2000s, incidence rates have either plateaued or continued to increase slightly. And so this really speaks to the fact that we are not doing a good job in terms of preventing breast cancer. And so that's really the motivation for a lot of my work, is to better understand risk factors and think about how we can prevent future breast cancers. And just in contrast, these are the rates of cardiovascular disease in the United States. And you can see since the 1950s, rapid declines in incidence rates of cardiovascular disease. So just taking a very big picture view, trying to contrast breast cancer to cardiovascular disease, why is it that cardiovascular disease has seen this great success in reducing the risk of cardiovascular disease? I think there are some steps that we as scientists, as epidemiologists, need to undertake to really think through how we can reduce incidence rates of breast cancer. And the first really comes down to research and understanding the science, understanding the mechanisms by which risk factors can influence disease. And then using that information, can we identify those populations that are really at the highest risk and target them either for actionable modifiable risk factors, such as for cardiovascular disease, it was stop smoking, exercise. Are there pharmacological interventions that do not introduce toxicity? And for breast cancer, I think we're still at that first box. I think we do understand a lot of the science, but identifying those at highest risk, we're still in the early phases. I think we are getting there, and I'm going to show you some of our work, but I don't think we are nearly as far along as the cardiovascular disease community. So now I'm going to shift a little and take a little time to describe the nurses' health studies. These are a series of studies. These are large scale, they're all ongoing population studies in which we enrolled large groups of women who, at least in Nurses 1, were nurses, registered nurses in the United States. Nurses 1 started in 1976. When 120,000 registered nurses agreed to participate in this study. They continue to be followed up today. Every 2 years, we follow up with a questionnaire. We ask them about their lifestyle factors. We ask them about risk factors. We ask them if they've been diagnosed with any diseases. And we, over time, as you can see in this picture, have collected different biospecimens, toenails, blood samples, urine, saliva, which has really allowed us to really examine a number of different disease exposure and disease relationships. And all of my talk today will be really focused on breast cancer. Nurses 2 is a very similar study. It was started in 1989. Recognizing that early life influences are really important for breast cancer. The enrollment at baseline, we made an earlier window. So we started really enrolling women as early as the age of 25 to 42. Most recently, there's a Nurses 3. This is an ongoing study that's still open to enrollment if there's anyone who's interested in enrolling. Again, recognizing how important early life is, we again sort of open the window to enrollees all the way down to the age of 19, and also enrolled male nurses as well. So just taking a little bit closer look into some of the study design issues with respect to some of the work that I'll be showing. Here I'm focusing on Nurses Health Study 2. Again, this study started in 1989 with over 116,000 nurses enrolled between the ages of 25 and 42 at baseline. One of the things that we did was that we collected blood samples from about a third of the entire cohort was willing to provide a blood sample. We collected blood from this large subcohort of women, and then we continue to follow these women to see who develops this disease. And this allows us to examine their blood sample prior to any disease to really see, do these certain molecular markers put individuals at a higher or lower risk of cancer. One of the areas that I'll be talking about and that I'm very interested in is using mammographic images to be able to predict later risk of breast cancer. And so we targeted our mammogram collection to the same time that women provided a blood sample so that we could also do correlative work to see if there were factors in the blood that predicted either breast density or other imaging factors. We also collected tumor blocks. So many, when people are diagnosed with tumors, many of their samples will be archived in Paraffin, and we were able to go back to pathology departments and retrieve those pathology samples and use that information to look at molecular characteristics of their tumors. And then we also were able to do a second blood collection in nurses too, 10 years after the first blood collection. And really what this allows us to do is to do some integrative epidemiology. So to be able to look at how the DNA may impact the mammographic features, or how their plasma metabolomics may increase their risk of developing cancer. And for each of these, we have subsets of participants that may provide their DNA or may provide imaging. And on all the women, we do have information on their lifestyle data. We also know where people live, and we're able to use geographic information systems to also collect environmental data. I'll just quickly highlight the guts cohort, which I did not mention before, but the guts cohort are the children of nurses too. And so when they were initially enrolled, they were between the ages of 9 and 17, whether they were in guts 1 or guts 2. We continue to follow them up today, and this really allows us to really look at, again, these early life influences and how that may impact later risk of disease. And you can see here that these individuals, as of today, are still mostly under the age of 40. So we're unable to look at breast cancer as an outcome, but I'm going to talk about some intermediate outcomes that may help us to understand breast cancer etiology. So now I'm going to shift to my first research finding. I'm going to talk about early life body size and breast cancer. So I think it's important to recognize that even though we think of breast cancer as a disease of adults, there are a lot of risk factors that we know of that are established that are happening much earlier in life, even as early as birth and in utero. So we know that birth size is positively associated with breast cancer. We also know that markers of puberty, such as menarche, is associated with breast cancer. We also know exposures and their timing are important. So we know that radiation, especially during adolescence or early life, is a risk factor for breast cancer, and it's much stronger if that exposure is early in life as opposed to later in life with respect to its impact on breast cancer risk. And you can see throughout the life course, there are different risk factors that are associated with breast cancer. And I think especially when we think about breast, there are different periods in life where the breast actually may be more vulnerable to specific exposure. So the breast is undergoing mammary gland changes all the way from gestation through puberty, when women go through pregnancy and lactation. These are all potential windows of physiologic change in which exposures may act differently in the breast than other periods of time. And, I like this figure here, which kind of shows on the one hand, the boxes you can see are sort of the life course stages. And then the white line above kind of goes through the course of cancer, how we go from normal to hyperplasia, dysplasia, eventually invasive cancer. And so you can see it's challenging to study early childhood factors as that may relate to invasive cancer. But one thing that we've tried to do is try and narrow that distance by looking at intermediate markers of risk, such as hyperplasia, or even understanding molecular expression in the normal breast to try and really understand how some of these factors may influence etiology of breast cancer. So one way that we study early life body size in epidemiologic studies is to use figures. So these are pictograms from 1 to 9, and we ask individuals to recall their own body shape, body size at different times during their life. And even though this seems sort of crude, and there's actually a fairly good correlation when we had people recall it and when we actually had measured BMI. Certainly not perfect, but it actually does a relatively good job. And in the Nurses' Health Study, we asked individuals to recall their this 9-level pictogram at childhood, during adolescence, and also at age 20. And what we found was a strong inverse relationship. So, women who reported that they were heavier or larger in size when they were in adolescence had a reduced risk of developing breast cancer later in life. And we also looked at different molecular subtypes of breast cancer. And what was interesting is that we saw equally strong associations for those subtypes that are hormone receptor positive, and those subtypes that are hormone receptor negative. And just to point out that this is actually a fairly strong risk factor when we think about sort of where this fits in, or protective factor, depending on how you want to think about it. But the association in terms of its magnitude is quite strong relative to some of our other established risk factors. And I just want to share these, we are not the only study that has seen this. This is generally been seen in many other studies all over the world and in different racial and ethnic groups, and a fairly similar magnitude of association. You can see these are just 4 other studies that have been published. They may have looked at BMI at different ages, whether it was 14 or 20. But you can see in general this sort of strong inverse relationship that girls that were heavier during this early life period had a reduced risk of developing breast cancer. One other thing that we tried to do, again, in most of those studies, it's either recalled, or actually in some of those studies, they were able to get records from childhood records. We were able to prospectively look in that guts cohort, so the children of nurses too, we were able to look to see if their BMI that was recorded when they were 10 was associated with, I mentioned this early life risk, or I'm sorry, earlier intermediate markers of risk. We looked at benign breast disease. So this is a early marker of later risk of breast cancer. And we did see that among girls who were heavy when they were age 10 had a reduced risk of developing this benign breast disease later in life. Again, kind of confirming in a prospective way that early life body size was associated with a reduced risk of this early lesion. We also looked at mammographic density. We were able to, which is a risk factor for breast cancer. And I'm going to spend the next part telling you a little bit more about mammographic density, but if you can believe me right now that mammographic density is a risk factor for breast cancer, you can see here that we see a very similar trend. What we see on the left panel is looking at the relationship between these pictograms and mammographic density, and on the right panel is the pictograms and risk of breast cancer. And again, you can see they parallel each other very similarly. And obviously, we can't make public health recommendations that individuals should be heavy early in life. And so really our goal is to try and better understand what is the mechanism that having a larger body size early in life may be important, and can we somehow translate that into a preventative measure. One of the other things that we did was, as you recall, we collected tissue samples from women who did develop breast cancer, and we were able to look at the gene expression profiling in their tumor tissues. And what we found was that there were different molecular profiles for cases if dependent on their adolescent body size. So we found that if we looked at both ER positive and ER negative breast cancers, larger adolescent body size was inversely associated with proliferative pathways. We also saw this especially for the ER negatives, which I think is especially important because ER negative breast cancer is a more aggressive subtype of breast cancer, and we've identified fewer risk factors. So we know that this body size, early life body size, is inversely associated with ER negative breast cancer and seems to be operating through reducing proliferative pathways as well as immune response and inflammation. The final piece of evidence that we have, really supporting this inverse association comes from GWAS studies. So these are genome-wide association studies in which we look at genetic markers and genetic scores for different exposures and look at what that association is with breast cancer. And this was work done by Chi Gao, showing that a genetic score for childhood BMI was inversely associated with postmenopausal breast cancer risk. So I think together this provides robust evidence that early life adiposity is inversely related to breast cancer risk. we see the strong evidence with these intermediate markers. We took this multidisciplinary approach with complementary data. Of course, again, the real goal of this is not that we would come up with public health recommendations to change people's BMI early in life, but really to think about, are there ways that we can harness this information and use that to develop prevention strategies. I think this also demonstrates how important that we consider the life course when we're thinking about risk accumulation, windows of susceptibility, especially when we're thinking about breast cancer. I'm now going to switch to mammographic density. So mammographic density, we often refer to percent mammographic density, which refers to the percent on a mammogram, the percent of the image that is occupied by dense tissue. So on a mammogram, if you look at the one closest to the left, which says none, there's very little white tissue. The white tissue is the dense tissue, and the dark on the image is fat. And so you can see that this represents increasing categories of percent breast density on a mammographic image, and the bars below it represent the risk associated with that. So someone all the way to the right who has more than 75% of their breast area on that mammographic image occupied by dense tissue has about a 5-fold risk of developing breast cancer compared to the woman all the way on the left who has a primarily fatty breast. So just a few things about mammographic density. It is an extremely strong predictor of breast cancer risk. Many of the breast cancer risk factors that are established are also associated with breast density in a similar direction. Breast density also tends to track well over time. So women tend, if they are high premenopausally, even though your breast density may change over time, if they're high premenopausally, they tend to be high postmenopausally. There is some evidence that you can change your breast density and that those changes are associated with risk. So we have in many studies used mammographic density as an intermediate marker of breast cancer risk. One of the first studies that I did with respect to mammographic density was looking at circulating estradiol. There had been this hypothesis in the literature that mammographic density must represent a woman's exposure to estrogens, and that's why it's such a strong risk factor and why we might think of it as an intermediate marker of risk. We were able to, in the Nurses' Health Study, because we collected mammograms at the same time that we collected blood samples, we were able to see, is there an association between circulating estradiol levels and mammographic density? We did not find a cross-sectional association between circulating estradiol and mammographic density. This analysis that I'm showing on this graph here, if you look at the left-hand panel, is looking at estradiol levels. And what you can see is that if you look at the red boxes, you can see that the increasing estradiol levels are associated with increasing risk of breast cancer. So in that 4th quartile, women with high estradiol levels have about a 2.5-fold increased risk of developing breast cancer compared to women in that lowest quartile. When we account for mammographic density, it really makes no difference, suggesting that they're independent associations. We did the converse on the right. So here on the right graph, we're looking at quartiles of mammographic density. This just also shows you what a strong risk factor mammographic density is. If you look at women in the highest quartile in that red box, they have an almost 4-fold risk of developing breast cancer compared to women in the lowest. And when we adjusted for estradiol, it really makes no difference, again, suggesting that they're independent risk factors for breast cancer. I will note that these analyses were done in postmenopausal women, so it certainly does not preclude that perhaps earlier in life, women's early life estradiol levels may be associated with mammographic density. Another analysis that we did was to look at percent mammographic density and later risk of breast cancer and to see was it associated with ER positive or ER negative subtypes of breast cancer. And we found that mammographic density was strongly associated with both ER positive and ER negative tumors. In this particular analysis, we are showing that it's actually stronger for ER negative breast tumors, but I think that what's important is that it's a strong risk factor for both. And I think this also speaks to the fact that it is likely independent of estrogen-mediated pathways. In this next analysis, what we did was we cross-classified people, really tried to ask the question, what if a woman had high estradiol levels and high mammographic density? And what we found is that if she had both of these things, especially if you focus on the women that never used exogenous hormones, those women were at a particularly high risk of breast cancer compared to women who had low estradiol and low mammographic density. We did a similar analysis here, looking at polygenic risk scores. So these are genetic polymorphisms that have been demonstrated in genome-wide association studies to be associated with breast cancer risk and are also very strong predictors of breast cancer. And again, you can see we did quintiles of polygenic risk score and quartiles of percent breast density here. And you can see there's a really, spreading out of risk so that women who have high genetic risk and high mammographic density are at a much higher risk compared to women who may be low on both of those factors. And I think this is really important because both of these factors, the polygenic risk score and mammographic density, have been helping us to improve our risk prediction models. This is work that we did in the Nurses' Health Study, and some of you may be familiar with the Gale model. There are several risk prediction models. I think the Gale model and the Tyrer-Cuzick model are two models that are used more regularly within the clinics, but they really they do a good job of predicting how many breast cancers there might be in a population. They don't really do a great job of discriminating, which of two women is really the woman that's most likely to get breast cancer. And we use this AUC metric to kind of get at that discrimination. And if the AUC was 50, it means it's as good as flipping a coin. And so, the original Gale model in our population was 55, so really not that great. But you can see with the addition of a polygenic risk score, the addition of mammographic density, the addition of circulating hormone levels, all of these things together are actually helping to move this away from sort of the 50 and actually to a point where it would be performing well in the clinics to identify those women at the highest risk. So to conclude the mammographic density section, mammographic density is a strong predictor of breast cancer. Mammographic density, and I didn't get time to talk about it today, but other imaging features that we've identified are associated with both ER positive and ER negative disease. Some of our other work is really addressing some of these novel imaging features, and they really do predict, increase our ability to predict breast cancer risk. And so, I think we will be moving to risk prediction models that include more than just traditional questionnaire-based factors that will be incorporating biomarkers and imaging, I think really is the future. And the final research vignette I would like to share with you is the impact of vitamin D supplementation on breast tissue. So there's several lines of evidence that suggest that vitamin D may play a role in breast carcinogenesis. We know this from cell line models and animal models, and it seems to be associated with both reducing risk of ER positive and ER negative breast cancer. Unfortunately, the epidemiologic evidence has been largely inconclusive, mostly showing null associations or an inverse association, but only within specific subgroups. These are data from a meta-analysis, and really, if you focus on the right-hand panel, these are the nested case control studies. So those are studies in which the blood was sampled prior to any disease, and it really shows very modest inverse association with breast cancer. So we were really interested in trying to better understand the impact of vitamin D on breast cancer and breast health. And we leveraged data from the VITAL trial. And this is a randomized control trial of high dose vitamin D. It's a 2x2 factorial design, which means that this was a study of men and women, were randomized to vitamin D and omega-3 fatty acids or placebo for either one of those. And the parent study was really focused on understanding the impact of high dose vitamin D and omega-3 on cancer and heart disease. These were the main findings from the study. They did not see an association with breast cancer. There was some suggestive evidence that perhaps death from cancer, and this is death from all cancers, may be inversely associated with randomization to vitamin D. So our goal, really our rationale, was that we felt the data with respect to breast cancer risk was null or inconclusive. There is some suggestive evidence that vitamin D may be associated with improved breast cancer survival. And it's really important to note that VITAL was not powered to specifically examine breast cancer risk. And really, given the long latency for breast cancer, which is sort of on the order of 15 to 20 years, unlikely that we would see an impact on breast cancer during the 5 years of active treatment. Our objective was really to see if we could see an impact of high-dose vitamin D on mammographic density and texture features, as well as an impact on gene expression patterns in breast tissue. So we conducted an ancillary study to this VITAL trial, and we collected mammograms prior to randomization, as well as post-randomization, 1 year after randomization, 3 years after randomization, and 4 years after randomization. And we were able to collect mammograms on over 3,000 women with, on average, more than 4 mammogram time points per individual woman. These are the main results of that study. And just to orient you, we are looking at the percent change in their percent mammographic density. The yellow bubbles are those that were women that were randomized to vitamin D, and the blue bubbles are women that were randomized to placebo. And what you can see is we actually don't really see much of an impact on you see that everyone is reducing, and this is expected as women age, their percent mammographic density does decrease, but we don't really see a difference between those randomized to vitamin D and those randomized to placebo. What is interesting is that we do see a difference in this V texture measure, and this V texture measure may be a little bit more sensitive of a measure relative to percent mammographic density. And so you can see that the line is a little bit steeper for vitamin D. The yellow bars have a steeper decline than what you see with the blue bubbles, suggesting that vitamin D supplementation may have a stronger impact in reducing this V measure, which is associated with breast cancer more so than in the women on placebo. The other thing that we were able to do is that we collected from the women in this VITAL trial, because we followed them up and we asked them if they had been diagnosed with a benign breast disease or breast cancer, we collected those archival tissue specimens. And we conducted gene expression profiling using RNA-seq. Dr. Chung Peng conducted gene set enrichment analysis with the cancer hallmark genes. The other thing that she did was she developed a vitamin D score. So looking at the people that were randomized to vitamin D versus not vitamin D, created a score, and then we tested that in an external population to be able to look at the impact on breast cancer survival. And what we found was a number, the red dotted line signifies FDR significance. And what we found was a number of gene sets that were downregulated in the women that were randomized to high dose vitamin D. So we saw significant downregulation of specific pathways, including proliferative pathways, cell cycle related pathways, inflammation, and all of these models were adjusted for age and ER status as well as stage. So in that second analysis that I mentioned, Chung created a vitamin D score, which tried to create a gene expression signature based on the women that were randomized to high dose vitamin D relative to not. And what we found on the left-hand panel is this gene score. We divided it up into tertiles and looked to see how the gene expression score was related to breast cancer recurrence. And we found that those that had a high vitamin D score had a reduced risk of developing recurrence. And you can see on the right-hand panel a very similar analysis, but using Metabrick. And it's very consistent with respect to both studies finding an inverse association with this high vitamin D score in a very similar magnitude. So to conclude the vitamin D portion, we found that high-dose vitamin D was not associated with significant declines in percent mammographic density. However, we did find suggestive and supportive evidence that high-dose vitamin D may have a stronger impact on V compared with placebo. And this may be due to the fact that V may be a slightly more sensitive measure of what is happening in the breast. We also found evidence that high-dose vitamin D supplementation influenced breast tumor gene expression profiles, and creating this vitamin D signature was associated with improved survival in independent study populations. So I think we're still somewhere between 1 and 2. I think we, understand more of the science than we ever have. And I do think we are getting really close into identifying high-risk populations to the point where we could make some actionable changes. Unfortunately, I think we're still, I think point 3 and point 4 are areas which a lot of research is being done and really where we need to focus our next efforts. So just to conclude everything that we've discussed today, there is strong and robust evidence, not just from the work I've presented, but from other work, that early life adiposity is inversely associated with breast cancer, evidence that novel imaging features can improve breast cancer risk prediction. There is suggestive evidence that high-dose vitamin D supplementation may improve breast cancer outcomes. And really, I think, the next steps are for future work to really better identify these modifiable factors so that we can come up with actionable recommendations for the prevention and improved survival of women from breast cancer. With that, I would like to acknowledge so many people go into doing this kind of research. I have a fantastic set of collaborators listed here. And I think very importantly, the participants that participate in breast cancer research and all kinds of epidemiologic research, specifically here, the Nurses Health Study participants and the Vital Study participants who have contributed their time, their effort, their samples. Really, none of this research would be doable without their efforts. And with that, I thank you. And if anyone has any questions, please do reach out. I've left my email here, and I'm happy to talk with anyone who's interested. Thank you again for your attention.